In this video we will discuss about cystic fibrosis and what are the different kinds of cystic fibrosis uh, occurs within the patient. So let us understand the cystic fibrosis, the protein that is responsible for the cystic fibrosis function. So it is actually a transmembrane protein that has different domains available. This is the membrane spanning domain 1, this is the membrane spanning domain 2, and it also has the few domains in the lumen, in the cytosol of the cell. That is nucleotide binding domain 2, nucleotide binding domain 3, regulatory domain, and this another domain at the end terminal end terminal carboxyl terminal at the carboxyl terminal, which contains a sequence of TRL amino acid, that is tyrosine, arginine, and leucine. As this sequence will play important role in enhancing the signals such as ion channels. It promotes the ion channel, that is to support the chlorine exchange within the within and outside the within and outside the cells. So what are the problems that occurs in the cystic fibrosis patient which are suffering from cystic fibrosis? Patients are not able to maintain or balance not able to maintain a proper balance between salt and water, and it mainly affects the cells that produce mucus, sweet, and digestive juices. Let us understand what are the different kind of defects that can be produced uh, in the cystic fibrosis patient, or what are the different kinds of cystic fibrosis present exist. First one is the considered as class 1, where this cystic fibrosis is not able to uh, transcribe translate transcribe and translate. This could be because of gene silencing or any other reason. Second one is considered as premature degradation. uh, this can be occurs because of the problem, or it is because of the problem in the structure, or gets because of the problem in the structure, and since the native structure, the errate mechanism directs towards proteasome for degradation. The third one is the class 3. In class 3, what happens is that there is a defect in the uh, ATP binding domain. So because of this, the CFTR will not be able to perform its function, as its main function depends on activation by kinase A, and that cannot be performed efficiently as there is a defect in ATP binding domain. Fourth one is the class 4, where it not be able to or it is not been able to translate or transport calcium ion from outside the chlorine ion, not calcium ion, chlorine ion from in and outside the cells, from outside in class 4 CFTR defect. It is not been able to transport chlorine ion from inside the cells to the outside to the extracellular matrix. In the class 4 defect, what happens is that the CFTR are been uh, CFTR not been able to express at the cell surface level, and the cell membrane uh, accelerated the turnover of this CFTR membrane. So this will create a less amount of CFTR present at the cell membrane. Fourth one is the last, that is class 5. Fifth one is the class 5 CFTR defect. What happens in this case is that the in class 5 defect of cystic fibrosis, the translation rate of this CFTR protein is low, or it is below the normal level. Because of this, the amount of CFTR protein required within the cell is not been properly produced by the cells. So let us understand one of the defects, or one of the cause of this defect. One of the cause could be the miRNA regulation of CFTR. miRNA means microRNA. So what happens in this case is that sometimes this microRNA bind to the 3' untranslated region, 3' UTR of the CFTR gene. Because of this, the translation of CFTR could not be able to complete, and this prevent or inhibit the formation of translation of CFTR protein from the RNA, and thus CFTR protein will not be able to translate normally. And another thing that promote the CFTR mRNA expression is that when the microRNA bind to the CFTR suppressor, thus thus inhibit the CFTR suppressor. It will not be able to interact with the CFTR promoter, CFTR mRNA, and in this case, mRNA can be translated at a higher rate compared to a condition where the CFTR suppressor is not inhibited, and thus in this case microRNA promote the growth of CFTR protein. So this is how microRNA can be used to regulate CFTR. So sometimes this because of microRNA could be the cause for the defect that has been present within the patient, and sometimes this microRNA regulation can be done artificially in order to get the outcome that has been designed by this microRNA regulation process. That's all for this video. Thank you for your time.